The event tonight has been put together by the Australian Institute of Food Science and Technology, the AIFST, with the support of QFI. AIFST, does everybody know about the AIFST? Have you heard about it before? No? Okay. Well, this is probably good to talk to you about. The AIFST is a membership association that provides support for anyone in the food industry and particularly supports students through the mentoring program that we have. Student programs like the summer school and also the student product development competition. Do the students here know about those? Yeah? If you don't, I'll be here afterwards and I'd love to have a chat to you about it. I also suggest that you head to the website as well. I can't tell you all about it tonight, but if you head to the website, there's heaps of details there as well. And also I'll be here afterwards and I'm happy to talk to anyone about the AIFST and what we can offer to our members. Just so everyone knows, tonight's presentation will be recorded and is available online as well. It'll be available via the AIFST website, but also the QFI website as well. So if you miss anything, don't worry, it's there. I would also just like to quickly thank the Queensland Alliance of Agriculture and Food Innovation for their generous support and involvement in tonight's event. You've done a fantastic job on advertising and just setting up. Thank you very much. I'd also like to thank the AIFST. It was organised by a gentleman called Giles Ailey, who works down in the office in Sydney, and so he's done it all remotely, so thank you very much to him. So just to provide some background on tonight's speakers, first we will hear from Mike Gidley. Mike Gidley, if you'd just like to put your hand up there. He's the Director for the Centre of Nutrition and Food Science here at UQ. The centre is part of QFI in conjunction with the Queensland Government, although I'm sure lots of you know about this. How many Mike Gidley supporters are there out there? Just a few? Yeah, they do. So Professor Gidley's research is focusing on structure, function relationships in biopolymer assemblies, such as starch granules and plant cell walls. This has led to the detailed characterisation of starch and dietary fibre digestion, fermentation in vitro and in vivo, with the understanding generated leading to opportunities for optimising nutritional value of foods and feeds. He is also a Chief Investigator in the Australian Research Council Centre and Excellence in Plant Cell Walls. We also have tonight the Honourable Mark Turner. Dr. Mark Turner is a tenured Associate Professor in Food Microbiology in the School of Agriculture and Food Science at UQ. Since completing his PhD at QUT, he has undertaken postdoctoral training in the USA and at QUT, and accepted an ongoing teaching and research position at UQ in 2007 and was promoted to Senior Lecturer in 2011 and an Associate Professor in 2015. He leads a research team in the area of food quality and safety with current grants with the Australian Research Council, Research Hub and ARC Industry Transformation Training Scheme Program and Horticultural Innovation Australia. He was the recipient of the 2017 Australian Institute of Food Science and Technology Keith Farrier Award of Merit. Welcome, Mark. So we might just leave question time till the end. We also have some food for you, which will come at about 7:30. But I might pass it now over to Mike.
Okay. Thanks very much, Vaughan, and good evening, everybody. It's good to see some familiar faces as well as a few unfamiliar faces, which is even better. So apologies to those of you who heard some of this before. I'm going to sort of start very broad and sort of work down towards the topic of prebiotics. So I've got a subtitle, Probiotics Revisited. And Mark's talk will have a lot more detail around individual issues around microbiota and health. So we're hoping that we can get this thing working together. Yep. All right, let's try that. Yeah, okay. So what I'm going to cover is starting off with what we understand about what's good for us in our diet, so dietary guidelines. and trying to work through from the dietary guidelines to an understanding of the role of the gut microbiota, and then get into some sort of where the food meets the microbiota. So talk about actually what part of our food that we ingest actually gets to the large intestine. How do we work that out? How do we work out what actually happens to that residual digester when it gets to the large intestine? And particularly focusing on the effects of the large scale, the architectural features of foods. So much of nutrition in the past has focused on the molecules of food, but we're in the era now where I think the focus is switching away from individual molecules and refocusing on the whole food which we actually eat. And then talk about some nutrition and health implications. So if you go to the dietary guidelines, there's a bewildering array of these around the world. The Australian one on the left there is a bit old-fashioned actually, but I still like it. I like the idea of the moving pyramid. I think that's particularly well captured in that one. And of course, in terms of health outcomes, it's not just what we eat, it's our various other aspects of lifestyle, particularly movement. More though these days, the dietary guidelines are expressed in the form of a plate. And so what you see there in the top right is the US version, the bottom right is the UK version. And here we're talking around not specific ingredients, we're talking about food types. So the US one is particularly clear and clean: fruits, grains, vegetables, protein, so meat or a vegetarian alternative, and dairy. The Australian and the UK ones a little bit more detailed and sort of somehow miss the point. The whole point is captured very well, I think, in the US one. So that's, you can ask the question, is this accepted around the world? And the answer is pretty much yes. So look at that US version and then look in the next slide at the Chinese version at the bottom left. There's really not that much difference. So you've swapped the plastic knife for the plastic chopsticks and spoon. You swapped the glass of milk for a cup of tea, but pretty much everything else is the same. And so this really does reflect a consensus that's developed around the world around what constitutes a healthy diet. Some people explain this in a much more sophisticated way. The Japanese one, I'm sure you're all looking at and trying to work out where they get to dietary guidelines there. This is very zen, but it really captures, again, the idea of balance. So in Japanese culture, balance is really important. And this idea of a top balancing, but balancing because it keeps moving, is a subtle but really quite a powerful one when you think about it. So you've got to keep the top spinning in order to keep it balanced. And to keep it spinning, you have to keep moving. So again, this integration of diet and exercise and how that all works together. But really what's underneath it in the essentially inverted pyramid is the same as in all the other things. So balance, variety, and whole, particularly plant-based foods. Different cultural contexts, but similar health consequences, positive health consequences. And that's really where our advice is now. So there's no advice out there now around how much protein, how much carbohydrate, how much fat. That's gone. That's history as far as the dietary guidelines are concerned. And so when we think around how we're going to look for foods in the future that have nutritional consequences, I would suggest we're going to look less at what the molecular constituents are and more about how much of the agricultural structure is retained. So that's a bit of my sort of way of thinking about it. In terms of where does those dietary guidelines come from? Almost entirely they've come from epidemiology. So that's where a large group of people around the world are continuously monitoring their food intake, food diaries, and the same people are also monitoring their health outcomes. And with enough people and enough events and a long enough time, then the statistics gets pretty powerful. But this is purely statistical correlation, and so it tells you nothing about how these things work. It just tells you that there are statistical associations. But I stress again, the numbers of people, more than 1 million people actively engaged in this all the time, in diverse environments, and over often many decades. This is a very powerful set of data that we have available to us. And that's where most of the public health advice comes from. The alternative way of doing nutritional trials is to have a very specific trial where you have a control and a test trial plus and minus a component. But where we're talking about the base diet of the grains, vegetables, fruits, you can't give a diet that's minus fruit or minus vegetables and add them. It doesn't make sense. So a lot of the focus on a more medical approach to nutrition has really been driven by these randomized controlled trials being the gold standard for nutrition. That's taken us down that road somewhat. So we need to rethink that as well. Okay, so if plant-based foods are the basis of a healthy diet, then we would argue then that the plant cellular structure, the plant cell walls, which we also call dietary fibre in nutritional terms, is the common component across all plant foods. And these are the foods which are typically underrepresented in modern diets, particularly vegetables, less so fruits, and particularly pulses, less so whole grains. What the epidemiology is pretty clearly showing is that the risks of developing many of the so-called non-communicable diseases, metabolic syndrome, diabetes, obesity, cardiovascular disease, colon cancer, are all fairly tightly controlled, so are reduced by the presence of a rich amount of fibre in the diet. Again, epidemiology is powerful here, but it doesn't tell you the mechanism. So the question in the science is turning into how do you bridge that gap between correlation and causation? Because with causation you can start to make changes and make predictions about the effects of those changes. And this is one of the areas where the gut microflora is a big focus of attention now. So there's some pretty reasonable evidence out there, and it's growing all the time, that the gut microflora are key mediators of the health benefits of plant-based foods, not just from the simple fermentation of dietary fibre, but so much more, and that's what I'll tell you about in the rest of the talk. So why do people get excited about the gut microbiome, microflora? So microflora are the organisms. The microbiome is the total genetic component that's present in the microflora. You hear a lot about this. A lot of the nutrition and health stories are around this now, and largely this is because of opportunity. So it's only been a few years that we've been properly able to characterize gut microflora, so particularly the DNA sequencing methods that now allow you to take a whole gut microflora, a fecal sample if you like, and just get a sequence of everything that's in there, and then disentangle that and say not only what microorganisms are there, but if you're lucky, exactly what activities those microorganisms have. This is incredibly powerful stuff, and it's getting stronger and stronger all the time. So this opportunity will be with us for quite a while, and the technology is still developing. The opportunity to harvest the information is still getting better. In particular, there's no longer a need for the microbes to be cultured and grown for them to be identified. So the microbes are identified by their DNA. They're not identified by growing them out and seeing what they look like, which was the classic approach. And that's also led to a sort of defocus of some of those bacteria that were able to be cultured in the past and which were the focus. So the lactobacilli, the bifidobacter, and so on. They had been studied a lot simply because it was convenient to do so, because they could be cultured in the lab. Many of the organisms that are present in our guts are very difficult to culture in the lab, but no more difficult than any other bacteria to be identified at the genetic level. We are a walking bag of microbes, basically. We have way more microbial cells than human cells. Of course, microbes are much smaller than human cells, so we're not physically more microbes than humans, but we have more microbial cells. And the diversity of microbial cells is phenomenal. And so in terms of the genetic diversity we're walking around with, most of it is microbial. And I think people are only really just starting to understand the consequences of that. So that much of our ability to deal with the world in a rapid and responsive way is driven by our microbes, because they can respond much faster than our human cells can. Microbes have typical generation times in the order of sometimes even hours, but typically days, whereas human cells turn over in several weeks to months. So our microbes respond much quicker to environment, including responding to food. So we're only really scratching the surface here about what the role of our microbes are for us, and in our context, our gut microflora in terms of how we handle our food. One of the obvious early starting points was to look at various gut dysfunctions, and now we know quite a lot more around the roles of microorganisms and the patterns that are associated with healthy and unhealthy gut functions, such as irritable bowel syndrome, diseases, ulcers, cancers, and so on. I think, though, the bigger excitement is coming that we're realizing the roles of the gut microflora go way beyond the digestive tract, so that they are associated with, and largely it's by association at the moment rather than true mechanism. They're associated with many good and bad parts of our health and wellness. So they're associated with cognitive function, they're associated with liver, kidney, heart disease, diabetes, and so on. And again, the correlative evidence for this is getting pretty strong. The mechanistic understanding is still in its early days. And so, as I said before, the gut microflora can respond quickly to diet and other environmental effects. And we can imagine that they're going to be the source of many biomarkers to sort of show what our status is. And so if we get into the stage of understanding what foods we might want to choose based on our current situation, you can imagine 5 to 10 years' time there will be a panel of biomarkers that will be based on our fecal outputs. And that's what you'll get when you go to the dietitian. In 5 years' time, will be my prediction. They'll all be reading out fecal biomarkers. Anyway, getting ahead of ourselves, let's go back to a few basics. And if we're talking about the microbes that are in our large intestine, we have to understand what of our ingested food actually gets there. And of course, we have 3 unit processes in our digestive process. We have a stomach where there's some digestion, but largely it's a holding tank of preparation and comminution of foods. Passes through to the small intestine, and at the top of the small intestine we secrete a whole load of enzymes that break down much of the protein, much of the triglycerides or the lipids, and much of the starch. And that is then the molecules that are produced from that, the fatty acids, the amino acids, and the glucose are then taken up in our small intestine, and that's where we get a large amount of our core nutrients from. What is not absorbed in the small intestine passes to the large intestine. So in terms of what gets the large intestine, it's a negative thing. It's anything that hasn't been absorbed earlier. And so it's not a positive thing. Apart from dietary fibre, which I'll explain in a minute, everything else is as a consequence of not being absorbed earlier. So what are the components? Well, the whole reason why dietary fibre is called that is because it is primarily carbohydrates, but it is not starchy carbohydrates, and we do not have the enzymes to break them down in our stomach or our small intestine. So there is no chance of us breaking down plant cell walls, fibres, before they get to the large intestine. We simply don't have the enzymes to do it. And then for a while it was considered that was the main aspect of what got to the large intestine. We now realize it's a lot more than that. Always there's some protein. If it's not residual from the diet, then it's secreted from, in terms of mucin proteins, down the small intestine. Those move through to the large intestine and provide a protein source. Certainly there is some starch, and we'll talk a little bit about resistant starch. It's when starch becomes fiber. Lipids, less known, but in some cases, certainly if you're eating lipids in nuts, then quite a lot of the lipids will get to the large intestine because the lipids are encapsulated in the fiber and they can't get out and be absorbed. Water and minerals. So water and minerals are taken up in the small intestine, but not all of it, and some of that gets to the large intestine. But it's also more than that as well. We're realizing that there are what have become known as co-passengers, so not only those things which automatically go to the large intestine, but those things which are shielded from digestion and absorption. So those, particularly in the context of plant foods, those are the micro and macronutrients that are trapped inside plant cells. So Sushil at the back there did this work. This is just showing if you have cells that you separate from legumes, this is chickpeas, then all the starch is still inside there, and even if you throw enzyme at it, which is coloured green here, the enzyme sticks on the outside, but it can't get into the cells. And so this is what happens. We think this is what happens in our small intestine, and so any starch from legumes that is present in a cell, we would predict would end up in the large intestine because intact cells are not hydrolyzed by the starch degrading enzymes. So even a single cell wall that's intact essentially is now carrying material through to our gut microflora. And this idea of now engineering our gut microflora through how we process our food is a reality, because we can choose how much we degrade down our legumes and our cereals and our other macronutrient containing foods, and we can expect to be able to control where they get digested and therefore what the response could be. It's not just those things which are trapped in the cells, though it's also things which are bound to cell walls. And so this is some apple cell wall material, and you can't tell it under normal light, but when you put fluorescent light you can see there are a lot of polyphenols associated with it. So these polyphenols, antioxidant polyphenols, are stuck to the cell wall, and they don't come off under the conditions of the stomach and the small intestine, and so these also get delivered to the large intestine. And so people who work on plant food diets now think this is an important part of the story as well, that it's not just the fibre that gets there, and it's not just the encapsulated macronutrients, but it's the bound up micronutrients, particularly the polyphenols. However, this is a difficult area because if you want to know what gets to your large intestine, you'd love to be able to sample at the gap between your small or the junction between your small and large intestine. You can't do that in regular people, not even students. So it really relies on people who've had their colon surgically removed. And of course, that's a very invasive thing to happen. And a lot of the work in this area has used ileostomy subjects. But there are real limitations because they have no colonic fermentation, and all the signals that we now realize are generated in the colon are no longer present in people who had an ileostomy. So this is a challenging area and will continue to be so, just actually working out what gets to our microbiota, what it is that they can feed on. It's not at all trivial to work that out. So what happens when you get there? So whatever the residual digester are and the secreta, particularly the residual enzymes and the mucin proteins, they're available for fermentation by our microflora. And there's a large number of different factors you could think about. So I'm going to focus more on the fairly simple things, really, which is related to how fast the fermentation occurs. That's got two consequences. One is that you don't want the fermentation to be too fast, because those of you who are unfortunate enough to be lactose intolerant know what that feels like when you have lactose. So lactose intolerance is where the lactose gets to the large intestine and there is fermentation there, and it produces gas very rapidly and gives all the discomfort. So we know that happens, and many people who have discomfort from various foods, it can be because they're fermenting things too quickly. We also would like things to ferment all the way through the large intestine, particularly in the context of colon cancer. Most colon cancers are at the end of the large intestine, and that's probably not surprising because then the fermentation, though, is known to be protective against colon cancer development. But if the fermentation has happened already, then there's no fermentation going on at the end of the large intestine. That's also not a good thing. So slow and steady fermentation is also a good thing to have. And so that's why I say the rate of the fermentation is related to the site of fermentation, because our food is naturally being pushed through all the time. End products, so there's a lot of talk around, and some of the health benefits from the microbiota are certainly due to the major end products of acetate, propionate, butyrate, all of which have good associations with health outcomes, and some of which become flavour of the month up and down depending on what's happening. But there's no doubt they have a positive outcome. And then the more detailed stuff around which microbes are promoted and what population shifts occur. So some key questions. Here's one for Deirdre. Who does what with whom and where? That is, which microbes work on which fibres and where does it happen and how does it happen? We should have a how in there as well. But we also have to look again at the architecture question. So there's the question of composition of the individual carbohydrates, but there's also the question of whether they're present as isolated, refined carbohydrates or present as an intact structure like a plant cell wall. And if you're talking about whole plant foods, you've got to think about particle size effects. So what's the effect of different sizes of bran or vegetables and so on. And then the consequences about micronutrients. I'm not going to talk about that, but that's an area that we need to do more work in. So some fairly old data now, I guess. It's getting for 8 or 9 years old from Barbara and Deirdre. Just showing you the time course. This is an in vitro. The time isn't meant to be the same as the time in our body. This is just a convenient time scale for us to work on. But showing several things. First of all, that some fermentable substrates go faster than others. And second, that even things that we think of as not particularly fermentable, like cotton, we should not think of anything as being completely unfermentable. Slowly, yes, but even cellulose can be fermented up to a point. I'm not saying it's fully fermented, but it's difficult to think of things which are zero fermented. And then some are fermented a bit faster, so things like bran have a reasonable fermentation profile. That's just to sort of as an orientation. The reason that we look at cellulose is that we try and build plant structures from the polymers upwards. So we have a system where we can put together the polymers that are present in a plant cell wall and make something that looks like a plant cell wall. So we can control its structure and we can then look at what the consequences of its degradation are. So we've done this for a range of common plant polymers. So we have 2 from cereals, arabinoxylan, mixed linkage glucan, or beta-glucan, are 2 cereal polymers. Xyloglucan is a polymer that's found in fruits and vegetables. And when they're present as soluble polymers, then they are fermented relatively rapidly, certainly much more rapidly than cellulose. But when we put them together with cellulose, they're fermented more slowly and more evenly over time. And the implication here is, and the composite profiles are similar to wheat bran, which is perhaps not too surprising because wheat bran is essentially a mixture of cellulose and arabinoxylan. So these sorts of profiles sort of give us, are the sorts that we're looking for, so that we have steady but the opportunity for complete fermentation over a reasonable time scale. And showing that it does matter whether the polymers are separate as a separate ingredient or whether they're present as a plant cell wall. Not only that, but the rate at which things ferment actually changes the end products that you get. And so what we're looking at here, just focusing particularly on the butyrate, because that's the one that showed the largest change, so that if you look at what happens with these individual polymers on their own, then you get these sorts of numbers. When you put it in the presence of cellulose, then you get double the amount. This won't always happen, but it's just to show there are differences in the end products depending not only on the chemistry, but in this case more particularly on whether they're together with cellulose or separate. So again, there's no additive way of doing this. You can't say, well, this polymer will lead to this amount of acetate and butyrate, and this polymer will lead to this amount, so we put them together, we can work it all out. No, it doesn't work like that, because the microbes work as a whole community on whatever they're given, and the consequence of that is not predictable from the individual components. So let's look at the particle size effects. So when other people looked at this for wheat bran, and it's been done a couple of times now, they got what we would have thought would be the logical thing, so that the larger pieces were fermented slower. And so you say, well, that's probably due to the surface area that's available for the microbes to attach to. But when we looked at pieces of carrot, we were surprised and thought we got it wrong for a while, but are now quite convinced we hadn't, where we found that the larger pieces were fermented faster than the smaller pieces. And that's what we're showing here. So sometimes this is where science gets fun, when things are thrown up that you're not quite expecting. But with the reason we think that that's happening, I hope you can see that, is that when you have these larger pieces, you've got a lot of cellular structure there, and this is actually a really nice home for the bacteria to grow in. So what we think is happening, that you're just providing a nice environment in which the bacteria can form a nice colony and can really start growing. If you've got very small particles there, the bacteria are trying to hold on to these small things and haven't really got haven't got the same sort of ability to be sheltered from the environment. So it's only a hypothesis, but it would seem when you have an open structure, maybe bigger is better in terms of promoting fermentation, whereas you have dense, maybe bigger leads to slower fermentation. So let's sort of summarize some of those effects of food structure. So nature provides us our nutrients in an encapsulated form. So we often talk in the food industry about encapsulation. We tend to mean that we've taken everything down to molecules and now want to build them back up again. But nature's already provided our capsules. We tend to break them too early. We have to be a bit more careful about that. So plant tissue foods, vegetables, fruits, cereals, and nuts, they're capsules. It's just that normally when we process them, we just break those capsules and we spend a lot of time and effort trying to put capsules back together again around them. The rate and site of fermentation question is I think an important one for formulation. And so it's known generally low molecular weight, more soluble carbohydrates are fermented more rapidly. Not only lactose intolerance, but some of the FODMAP type of area is important here as well. We know that dense and lignified structures are less fermented, particularly lignified ones, and that larger particle sizes can either lead to faster or slower, depending on whether they're open or condensed. And this brings us to another sort of need to rethink how we talk about things. So in the past, people tended to classify dietary fibres, well they still do, as either soluble or insoluble, and say, well, this is what soluble fibres do, this is what insoluble fibres do. Yeah, the problem is that almost everything that we eat is a mixture of everything, is a mixture of both. And that some soluble polymers are not fermented very well. This is how psyllium works. So psyllium is metamucil. This is how that works. It's essentially a soluble fibre, but because its chemistry is so complex, we haven't given our bugs enough time to work out how to degrade it, but give them enough time, they'll find a way. And some so-called insoluble polymers, particularly those from white flour, they're fermented just as quickly as the soluble polymers. So there's no real necessary relationship between the solubility of a polymer and how it behaves in fermentation. And we're working on how to better categorize fibres, because we see the limitations of soluble versus insoluble. So that's the food side. So what about the microbe side? Outside my area, and I'd be interested in people's comments. So my way of looking at it, we've at least got two rules of thumb which we can work with. One is that if you get a lot of protein getting to the large intestine without too much carbohydrate, that is almost certainly going to be a bad thing. And that is because if our microbes are fermenting protein, then proteins have a whole series of different chemistries in their side group, in their side chains, and the microbes will produce side products from those, particularly phenols, cresols, various amines, ammonia, and so on. And Mark's going to talk about one example of that. And if you don't have a lot of carbohydrates there for the bacteria to grow on, then this is what the bacteria will do. So this is the downside of having readily digestible carbohydrate along with your protein, that there's no carbohydrate to hold the hands of the protein when it gets to the large intestine. However, when carbohydrate fermentation is abundant, it may well be that getting protein to the large intestine is a good thing, because microbes have a lot of protein in them, they need the amino acids in order to build more bacteria. And so if you can actually provide some of those proteins through the diet, as long as they're busy fermenting carbohydrates, they can use that protein to make more bacteria. So again, it's not as simple as, it's not good versus bad, it's how the whole diet works is the key to this. The other general principle is about diversity and abundance, so that now that we can do this complete characterization of the microflora at the DNA level, we no longer focus on Bifidobacter and Lactobacilli as we had in the past. They would still be in the so-called good guys basket, but they're only a relatively small part of our total gut flora. The story is a lot more complex than that. I'll show you in a minute some of the sort of overview evidence, but lack of diversity. So this is essentially just how many different types of bacteria we've got. It's a very crude measure, but it's something that comes out relatively easily from the genetic analysis. And it's proving to be surprisingly good at predicting health status, so that lack of diversity is associated with a poor health status, increased diversity is associated with a good health status. Some of it's logical and obvious. If you've got more helpers along with you, there's more chance you can cope with what's coming. But really the mechanisms are hardly understood at all. And then there's the other principle that is less established but seems likely, is that a diversity of fermentable carbohydrate leads to a diversity of microbes. Now we're making a link back to the health advice that comes from epidemiology that says food diversity is important. So maybe part of the reason why food diversity is important is that microbial diversity is important. So just one example, this is getting a bit old now, but and there's a lot of more recent, more detailed work, but this was one of the first sort of large-scale studies that really oversimplified perhaps, but just so that you get the message, it came down to one cartoon, about 20 people years work down to one cartoon. So this was comparing cohorts who are either non-obese or obese. So working from the top, obese cohorts have a higher risk of metabolic disease. So metabolic disease is diabetes, cardiovascular disease, dyslipidemia, and so on. That's well understood. And the interesting thing was that that was in contrast to the complexity of the microflora, so that the obese individuals had much less microflora diversity than non-obese individuals. But the good news was that if that was treated with appropriate diets, then you could reverse that. And so it doesn't mean that they're reversing the metabolic syndrome effects, but you can change the microflora through diet, and you can increase the complexity with a suitably complex plant-based diet. It remains to be seen how far that can go towards treating pre-existing effects, but that's where a lot of work is going at the moment. So the fact that it responds to dietary intervention at the microbial level is really important, because that gives us the chance to make things happen through the diet. And so just one example from our work, from Lucas's PhD thesis. We work with pigs because we can then get samples from inside the digestive tract, so we can look at how things are moving along the tract. And here we're looking at the number of microbial species, operational taxonomic units. And so from what I've just said, you want this number to be big, because that tells you that there's more diversity present. And here our control was the red, so it's a control diet. The blue was when we added some mango pulp to the pig feed, so they loved that. And the yellow was when we added isolated pectin. So the main fibre in mango pulp is pectin. So when you added isolated pectin, you got a negative result in this context, in the diversity actually went down. They were very good. They produced a lot of microbes, but they were the microbes that were just working on pectin only, rather than a whole bunch of microbes that were working on not just the pectin, but all the other stuff that comes with the mango pulp. So there's a few other pieces of evidence for this as well. So this is another piece of evidence that changing the microbiota is about changing at the food level, not changing at the individual ingredient level. And so coming down to the end, so this is really saying, so prebiotics. So prebiotics I'll come on to the next one. So probiotics, here's the definition from 1995 when they first were proposed. And was a great concept, and so you can read the original definition there, and it's based on the ability to isolate individual organisms and to show that they've got more of them after you've had a dietary intervention, and the classic ones are the bifidobacter and to a lesser extent the lactobacilli. And so we can say one of a limited number of bacteria already resident in the colon. And that's what happens with our microbiota when we have different foods. It's not that we kill and magically create new microorganisms. It's just that the already wide population is either skewed towards a few or the whole is promoted. So this stood the test of time until the sequencing technologies came along. And so you can see this in the way that in the way that various food products had been advertised, that we can no longer really say this because we know that this is not enough evidence to, or just having a limited number of bacteria growing is on its own is only one part of the story. You need to look at the whole microflora as a community. And so, this was from Glenn Gibson at the University of Reading in the UK, and so he and a whole bunch of others got together a couple of years ago and tried to come up with what would a modern version of a probiotic be, and this is where they got to. It's slimmed down, always a good thing to make it a shorter thing, and yeah, look how broad it's got. And this recognises several things. It says, utilised by host microorganisms, so we're not putting new organisms in there, we're working with what's there, but we're tailoring, we're pushing it, reprofiling it, but we're conferring a health benefit. Wow, now that's a pretty broad statement, right? And so, but this really summarises very neatly, in my view, where we're at. We sort of know that there are a lot of health benefits to be had, but we can't put our finger on exactly what the cause and effect relationships are. Except that we know that there are important things that can be done there. So that brings us back to this, and really, I guess my point is to try and hammer home that if you're following these sorts of guidelines, you're also helping your microbiota, and your microbiota is a large part of the story about why this is what we are advised is a healthy diet, and that's the more that we learn about how particularly the plant foods are handled through the microbiota and the consequences that has for our system, the more that we will be able to be a bit more selective about how we put diets together at a dietetic level, and at an agriculture and food level, what we choose to grow and process and how we present food. So that's it. So pretty top level stuff, but I just wanted to try and show you how central the role of microbiota has become in our understanding of what a healthy diet is, but also how much remains to be done to really understand how you can make a difference through individual foods, meals, let alone whole diets. So I'm going to hand over to Mark now. We're going to do Q&A at the end. I guess if there's questions of clarification, I could take that now, but generally if we want to have a discussion at the end, we'll do that. So if there's anything you want to clarify explicitly, we'll do that now. Otherwise, I'll hand over to Mark.
All right. Well, thanks for coming. And so my name's Mark, and Vaughan gave a lovely introduction of me. And I have to say, Vaughan's a former Bachelor of Food Technology student at UQ, and I have to say he's one of my favourite students at UQ, Vaughan. I just wish you'd come to more lectures. I hardly ever saw Vaughan in the lectures, unfortunately. So he must have been busy at the time doing other things. I'm not quite sure. But it's great to see people like Vaughan, I guess, and UQ alumni, our former students, doing so well after they graduated and moving in their careers and doing great. So it's great to see Vaughan. I joke with Vaughan every time I see him. But we know we mess around. And I want to thank Mike for that really nice overview talk as well. And this is inspiration for me at the dinner table with my 10-year-old son, trying to get him to eat vegetables and so on. He loves McDonald's, but unfortunately when vegetables appear on his plate, there's a massive fight. And I continually tell him about the bacteria, and he's got to eat these vegetables for his gut bacteria and so on. So he's kind of sick of hearing all this sort of stuff, but I blame Mike for spreading that word. Anyway, I'll get to him sooner or later. So what I wanted to do is just kind of, my talk will be quite different to Mike's, and I really want to go into some detail on some recent specific studies and all the elegant work that's happening at the moment. Mike's been in this area a lot longer than me. I've read a lot of papers in the last 2 weeks. It's a bit of a cram session, I have to say, like the students before the final exam. But I really wanted to sort of showcase some of the really nice data that people are getting to really come to some of these conclusions about the role of diet impacting on gut microbes and how that's affecting our health in specific areas. So a lot of us have probably already seen this, and Mike touched on this as well, that we've got a bag of bacteria living inside us, and whether we're just a giant microbe walking around, or whether are they controlling us or we're controlling them, or what's happening. So we've got 10 times more bacteria in our guts and 100 times more genes from bacteria in our gut. But it's not just the gut bacteria, there's also evidence coming out that viruses that infect those bacteria are playing a role as well, and we kind of ignore that, and I think to our detriment, and I think that will probably be the next thing. So the bacteriophages, how they affect the gut bacteria and so on, how this affects our health as well. And I guess when I studied at university back last century, in microbiology and in the textbooks, it was kind of like, you know, there's some bacteria in your gut and there's E. coli that produces some vitamin K or something, and it doesn't really matter. It was quite irrelevant 20 years ago, but it's becoming a very different story these days. So first of all, I just wanted to talk about some specific areas about how our gut microbes might be communicating with us, and just moving beyond the gut, the effect those microbes have in our intestine, and how they can affect different organs in our body. How does that happen? They're distinct. Our brain is far away from our intestine. How can that communicate? There's evidence to suggest that the gut-brain axis exists and how this works. The second example I'm going to talk about is some, and Mike talked about this degradation of proteins to produce ammonia and nitrogen-based compounds, and how this is impacting on cardiovascular disease. I'll talk about some meat-munching gut bacteria and how they're working. Then I want to talk a little bit about some probiotics and what researchers are doing to looking at mining the microbiome. So when you characterise the microbiome, are there bacteria in there that are possibly, can we use these as the next generation of probiotics? I know there's a massive probiotic industry in Australia and overseas. A lot of these probiotics are really based around, you know, they're kind of, we found them in someone's gut or we found them in some fermented foods, and really they've just been sort of added into some yogurt and then marketed as, you know, having some probiotic health effects. So I guess there's a huge number of bacteria that we can look at, and if we've got evidence to suggest that they might be useful, then we can kind of look at developing new probiotic products based on that. So one example I've got here is you are what you or your mother eats. So this is not just about your diet, but what your mother ate before you. I want to talk about salt selection of gut microbes. And then last of all, I'm just going to talk about a sugar, Trealose, which is a novel food sold in Australia, and how it's impacting on infectious diseases. So these are the examples. So the first one, so how our gut microbes are talking to us. And there was a paper about 10 years ago where they started using metabolomics, which is the techniques involved in this are quite staggering, and the equipment and so on, where you can screen for thousands of compounds in samples like blood, and researchers were interested in using these techniques to basically look at what the role of the gut microbiota has on in our blood in compounds, chemicals that actually make their way into our bloodstream. So this suggests that this provides a link between the gut and other parts of the body through the blood. So they did a simple experiment where they basically looked at the metabolites in mice that are germ-free and also conventional. So the way they make germ-free mice is they give mice a dose of broad-spectrum antibiotics, which basically destroys all the bacteria in their gut, and then they house them in a sterile environment and only feed them sterilised food and so on. So it's very, very controlled. And then they have these conventional mice, which are just fed a normal diet and have a normal gut microflora and so on. So they just basically took blood from these two different groups of mice, and then looked at the metabolites inside the blood, and what they found was there was 3 times more compounds in the conventional or gut bacteria containing mice. So up here's the conventional mice, so they had about 145 compounds. The germ-free only had 52, and that combined they had a lot of compounds that were the same. So this suggests straight away that you've got some gut microbes that are impacting on the chemicals that are floating around in your bloodstream. And they also found that about 10% of the common compounds out of these 4,000, so around 400 compounds, there was quite a large difference in the level of those compounds as well between the germ-free mice and the mice with the gut bacteria. One of the biggest compound changes they saw, and I'm not going to go into this too much, is one of these indole-based compounds. And this is basically derived from an amino acid that we eat in our diet, tryptophan. And this is the gut bacteria here. So it's converting it into indole and then modifying it. And then this passes into the blood, so this is in the gastrointestinal tract, passes into the bloodstream, floats around in the blood, goes to the liver and can be converted into the liver into another compound. So this was kind of some nice evidence to suggest straight away that there's gut microbes are fermenting the food and producing compounds which are going into the bloodstream and then can move to other organs as well. And this makes sense, I guess, when you think about the gastrointestinal tract. The main function of it really is to absorb nutrients for energy and for our cells to divide. So anything that's in there will want to be absorbed into our bloodstream. So this transfer from the lumen of the gut into the bloodstream is not unusual. Okay, I want to move on to this meat-munching gut bacteria now. So atherosclerosis here is a disease, a cardiovascular disease, which is due to arteries narrowing due to the buildup of plaque, and this can lead to things like strokes and so on. And it's due to lipid accumulation and inflammation of the arteries in humans. So they found in the past, this has been known for many years, a high level of meat intake is associated with atherosclerosis. So they suggest that you don't eat too much meat because you might be prone to this disease. But they never really knew why. And a lot of studies were looking at, is it because of the fat intake, because meat has high levels of fat, is that the problem, and so on. But they could never really link this as such. And just the last few years, some groups have done some really nice studies to show that the gut microbes are actually an important, crucial part of this conversion of meat-based products into this disease. And so just to cut a long story short, I'll just show what's happening, and then I'll show you some of the data that they've got. So they've basically shown that red meat-rich diets or things like eggs and cheese and seafood have these chemicals, carnitine and choline from acetylcholine. And these are converted by the gut microflora after we consume these, and it's basically cleaved at this bond here, and you end up with this compound trimethylamine. This then is absorbed into the bloodstream, moves to the liver, and then it's converted. It puts a hydroxyl group on here, trimethylamine oxide, and then this is a major cause of atherosclerosis. So I wish I was working in this area because there's so many papers in Nature and Nature Medicine, all these sort of high-impact journals that I only dream of. But food microbiology journals are impact factor 3 and 4. These are 40 and 50 and so on. But anyway, it's a lot of work they do. It's really nice. So this trimethylamine oxide production requires the gut microflora. So how do they prove this? How do they show that first step is actually happening? So what they did was they took some humans and they fed them some steak and carnitine. That sounds like fun. I think 200 grams sirloin steak every day. Yep. And then they treated them with some antibiotics to flush out the gut microflora. So did that affect this trimethylamine oxide in their blood? And sure enough, it did. So the first person here, so the group here, you can see this trimethylamine being produced in the blood. Then they suppress the gut microflora, so they hit them with broad spectrum antibiotics, basically to just kill all the bacteria in their gut. That chemical disappeared. And then they took them off the antibiotic and then kept feeding them the steak every day and carnitine, and then the chemical reappeared again. So there's some really nice evidence to suggest that the gut microflora is important in converting this into this chemical here, which is linked to the disease. So the interesting thing, they also took some vegans or vegetarians. So they're obviously not familiar with meat and so on. They've been off it for a while. They managed to convince some vegans and vegetarians to eat some steak as part of research, which is quite impressive. So what they did there, this is the plasma levels of that chemical. So there's an omnivore here, so this is a meat and vegetable eater, so their gut microbiota is familiar with consuming meat, whereas the vegan here is not. So what they found is the trimethylamine oxide levels went right up when they were on meat, whereas the vegans didn't increase. So they were eating the steak and the carnitine and everything, but it didn't change. And this was basically suggesting that, they did some more as well, so they found a few more people. They thought that was really cool, so we'll get some more numbers, n equals 5, n equals 5. The omnivores, the level of the trimethylamine went up again, but the vegans and vegetarians didn't respond at all. So this suggests that the vegans and vegetarians don't have the gut bacteria that will actually metabolize that meat, that carnitine and choline, which makes sense because if you don't give them the food, they won't grow and proliferate in your gut. So really, preceding dietary habits modulate intestinal microbiota composition. So I don't know if you've kept feeding these vegetarians for months and months, they would probably find that bacteria somewhere and then start responding in such a way. When they look at the gut microbiota, they find differences in the populations. So this is looking at the microbiota of the vegetarians versus the meat eaters and so on, and the two different groups of bacteria here and corresponding with the levels of that chemical. And basically they find that the level of these two groups of bacteria were lower in the vegetarians as well. So they were trying to link the metabolism of those compounds with the two different groups and also the levels that they produced. But I'm not going to talk about that too much. So they've shown that high levels of this carnitine is linked with cardiovascular disease in humans, and with a large study 2,000 people. And so they wanted to establish this in a mice model as well. So these are aortic cross-sections showing plaque buildup, which is a model for atherosclerosis. And mice fed a normal diet here. This is what it looks like. You see some plaque there, but one's fed a carnitine diet, which is from the red meat and so on. You see there's much higher buildup of this plaque here, so that's showing the symptoms of this disease. So an easy experiment is just to give those, if you give that mice antibiotics and feed it the carnitine, do you see a reduction in these plaque and the disease? And sure enough, you do. You see it go from here to here. So that's again providing evidence to suggest that the gut microbiota are actually doing some metabolism that's actually impacting on these arteries and the plaque formation. They've done some more mechanistic stuff. I'm not going to go into it here because that paper was massive and it took me at least several hours to read. There's so many supplementary figures and so on. Oh my God. But basically they've shown that this chemical here is able to reduce cholesterol excretion in the bile by the feces. So it seems to signal that. I don't know how it's doing that. I'm sure the next Nature paper will probably reveal that in a few years' time. But it seems to be a part of the process of getting rid of this. The next sort of thing they did was just 2 years ago, was they're looking at, can they actually block this process in the gut? So you've got the gut microbiota there. Can we actually design a drug that will inhibit this chemical reaction to produce that TMAO, which causes the atherosclerosis? So they were looking at, can we drug the microbiome? So this is possibly a new, instead of giving you a drug to treat your disease directly, can we use a drug that actually blocks the microbes from metabolizing something? So it was kind of a novel concept, and I guess that's why it's in Cell. So they screened for an inhibitor of this TMA, which is the precursor to the TMAO, and they got this chemical here. So the idea was here that you'd have the choline or the carnitine. It gets digested in the gut by the bacteria to these chemicals in the liver here, and this leads to cardiovascular disease. So this chemical DMB here is an inhibitor for an enzyme that converts this to this. And you can see the structure is very similar. It's just got a carbon instead of that nitrogen in that spot there. And sure enough, when they did the trials in the mice, they fed them the choline here, so you can see the buildup of plaque in the arteries, and then when they added the inhibitor here, it disappeared. So they're sort of talking about this as a prevention and treatment for atherosclerosis. But I guess another way to do it is just to eat less red meat and things that actually have that. So I mean, all this elegant science that goes on, you sort of think, come on, there's a simpler way. Just sort of reduce the amount of this at the start, I guess. So anyway, that's very nice work. Just kind of, I guess, sort of start to provide some links between diet and health through the gut microbiota. All right, this next study is really interesting. So it's referring to things, the diet of your parents and how they might affect you. And this was all done in mice, so they didn't sort of modify humans as such. This is the study here. All right, so what they did was they took some mice and they fed them on a high-fat diet and just a normal diet. And then they were female mice, and then they bred those to produce some offspring. And they were characterising the offspring of those mice, fed the high-fat diet, so they would have been obese mice and then normal weight mice. And the reason they were doing this is because looking at the social behaviour of those, because there's evidence to suggest things like autism is related to, the rate of autism is related to the weight or obesity of mothers in humans. So they were looking at sort of, can they get a model for this sort of situation here? And autistic symptoms involve social behaviour, quite introverted and different sort of social behaviour. So they were looking at, can they recapitulate this study in mice? So what they did was they were looking at social behaviour between mice. It's much easier to study that than humans. So they get the little mice there and then they put them in a little box and then they record and see what they do with each other. Do they sit in the corners and just ignore each other? Or do they come and like sniff each other and move around and something like that? Anyway. And what they found was the offspring from the obese mother mice were less social. So these are some graphs here I'll show. This is just the mothers fed a normal diet, so their interaction time and contact duration. So these little two mice here are kind of hanging around each other and they're very social with each other. I don't know what mice do socially, but maybe they communicate by twitching their noses or something at each other. So these were the mothers were fed a normal diet. These ones were the mother was fed a high-fat diet. And these poor little mice here kind of just sat in the corners and didn't talk to each other as much. Here, the interaction time was lower and the contact duration was lower as well. What they found was when they co-housed these mice, from the mothers, such a long thing to say, from the mothers with a high-fat diet with the mothers with a normal diet. They put them together, so the two different types, and they thought, if we put them together and then do the test, do they change? And sure enough, they did. So these were the normal mice here, co-housed together, and they were interacting really well and contacting each other. This was the ones that the mothers were fed the high-fat diet, so they were less social here and here. But when they housed these guys with these guys, they found these guys, their social activity increased. And maybe because the normal fed mice from the mothers were teaching them how to be social. I don't know, mice maybe can't do that. But they thought, obviously, maybe the other thing is that they're sharing because they poop all through the cage and everything. They're eating each other's poo. They're sharing each other's microbiota. So maybe these guys here are transferring some microbiota into these guys and then improving their social skills. Sounds interesting, doesn't it? Yeah. Scientists have too much spare time on their hands, I think, don't they? So what they did then, that was their hypothesis, that the gut microbiota is different between these two groups of mice, and that explains why they're social. It's interesting. So they did the gut microbiome analysis, and as Mike said, this is really revolutionary in the last 5 years or so. It used to probably cost $1 million to do this kind of experiment, but now it's in the order of $50. You can get a huge amount of data, analyse thousands of bacteria just by sequencing their DNA. What they did was compared the gut microbiota profile, the different species between the two groups, and the number one bacteria that was different between them was called Lactobacillus reuteri. And yet the offspring from the high-fat diet mothers had 9 times less Lactobacillus reuteri than the other group. And this is a good bacteria because I did my PhD on this one a long time ago as well. So I'm happy to see it up there. It's actually a commercial probiotic as well, I'll have you know. I'm not getting paid to say that. So what they thought they'd do is say the antisocial bacteria have less of antisocial mice have less of this bacteria. So the idea is, can you supplement that bacteria, feed it to those mice and improve their social interaction? So that was the next experiment that they did. So here's some graph here showing social novelty, and here in this one here, there's some other data I'm not showing, but they get 2 mice and they put 2 mice together and then they add another mice, and then they ask, does this mice that they're testing, does it go to the new mouse, and is it like interested in that new mouse? Because normal mice they're inquisitive, there's a new mouse over there, I'm going to go over here and start talking to it. So the normal mice do that. So you see this increased bar here for the normal mice that the mother was fed a normal diet. For the mice where the mother was fed a high-fat diet, they don't do that. There's no difference between the old mice and the new mice. They don't care whether there's a new mice there, whatever. I'm just going to keep going to the old one. It's just, I'm not interested in anything. But when they fed that mice, they fed them 3 different things: just growth media, heat-killed Lactobacillus reuteri, and also live Lactobacillus reuteri. And the live Lactobacillus reuteri here led to an increase in the social interaction with this new mice as well. So this is really nice. It's quite a substantial change. Just providing that one bacterial species back into that mice triggered such a big difference in its social activity. It's quite staggering to see these results. Hard to believe sometimes, actually, considering the complexity of the microbiota. So how does this work? Lactobacillus reuteri has been known in some other work shown to increase oxytocin levels, and oxytocin is known as the love hormone. I found that on Wikipedia. It's a peptide that's produced, and it really is produced in your body and makes you feel like, I really sort of, I love doing this. It's a really enjoyable hormone, makes you feel like that. So it kind of makes sense that it would improve social interaction. You're more sort of, you're feeling good, you're feeling more connected with other people and so on. So that's quite interesting and makes sense. So what they did was they add some oxytocin into those mice that were struggling, and also it rescued these things as well. So the bacteria was probably producing something that was triggering the oxytocin levels as well. So my question is, in humans, this is all done in mice, so in humans, can probiotics improve your social life? So by taking Lactobacillus reuteri, do you have more friends on Facebook? I don't know. There might be some analysis someone can do there. Maybe drug dealers in the future, instead of providing love drugs, party drugs, ecstasy or whatever, I guess, maybe they'll provide putting capsules of Lactobacillus reuteri out at the things, That might be a lot safer. I don't know. I think some more data needs to be shown in humans rather than mice. Anyway, it's really nice stuff. All right, the next one I'm going to talk about is salty selection of gut microbes. And as we know, we all eat too much salt, and these are some numbers in the US, 8.5 grams per day in the US. And I just looked up the US because it was easy to find. It's probably Australian numbers, and we should be less than 5.8 grams of sodium chloride. And having too much salt can lead to high blood pressure. And I have to say, yesterday I was reading the Courier Mail, and there was a story about how sausages, the great Australian food, there's about half the amount of, half the daily dose of salt in sausages. And yeah, kids shouldn't eat sausages and everything like that. And this made me very disappointed, because one of my favourite things to do on a Saturday morning is to go down to Bunnings Warehouse. And I go in there to buy something, but I walk past the sausage sizzle, there's always one there, and it's just the smell just tracks me to it every time. So it's quite sad. So I want to know, is it possible that I can enjoy that sausage and have a higher salt dose and be able to do something with my gut microbiota that can allow me to handle that higher salt? And I think this study might provide an answer to that. All right, so this was a paper in 2015. So really looking at, does the gut microbiota play a role in this effect of high salt diet on our health? So again, they go back to mice, because humans are a bit harder, but they did do some human trials at the end I'll talk about. So what they did was they fed some mice a high salt diet and a low salt diet, and they looked at their gut microbiota. And what they found was the gut populations of lactobacillus were reduced the most. And Mike kind of mentioned lactobacillus and bifidobacillus are really least dominant, and that's true. So, you know, maybe 1% of the populations and so on. There's a lot of other organisms, Clostridia and so on, that are really dominating. But some of these studies are really sort of showing, I guess, that some of these really low abundant organisms seem to have some effect as well. So it's quite interesting. So the one species they found was reduced the most in the high salt diet one was this Lactobacillus murinus. And this is the abundance of this species, and you can see that it's 1% or less. It's about 0.5% of the total bacteria. So it's only 1 in 20 of the total bacteria that's there in these mice. And this species here is in mice only. It's not found in humans, which is a bit of a bummer for me, going to Bunnings and so on. But what they did was they had the mice on a normal salt diet, and the numbers of those Lactobacillus murinus were okay. And then this is every day. And then they hit them with the high salt diet, and it was pretty high. And the levels of this species here dropped very low during that phase of feeding, and then they return it to a normal salt diet. And the levels of those, there was obviously enough there to rebound back up to sort of normal levels and recover pretty quickly. So what they did was they got the mice and they got those little, you know, those blood pressure things. I don't know, they must have been very small to measure the blood pressure of the mice in their arms, I'm not sure. So they measured the blood pressure, the systolic and the diastolic here. So these are the numbers here. This is the high. When you measure your blood pressure, you have two numbers. The one where it's after it's pumped the blood is the high number, and when the heart's relaxed, it's the low number. I found that on Wikipedia as well. And obviously, the lower the numbers of these, the better off you are, unless it's 0 and 0, which means you're not in good shape anyway. So, but under normal salt diet, the level was here. Under the high salt diet, it went up as expected. We know that. When they put the mice on the high salt diet with the bacteria supplemented, it reduced a little bit, but it was significant. The other levels here, the diastolic here was much greater, so the normal salt was lower, high salt went up, and then when they added that bacteria, it reduced again. So this is quite interesting. It's suggesting that you can kind of add some sort of probiotic and to restore the levels of that bacteria because they reduced at the salt, and we can kind of get them back up, and then that will actually affect blood pressure. How does it do this? There's a mechanism that they present, and I talked about these indole metabolites before from tryptophan, and they've shown that the Lactobacillus murinus here this is in the gut, this is in the mucosa in the blood. So the bacteria metabolize tryptophan to produce these indoles under normal conditions when everything's happy, and this blocks the T lymphocytes, which is an immune white blood cell, to differentiate into this other type. I'm not going to go into this. Immunology is way beyond me. So this is in a normal diet. In a high salt diet, it knocks out that Lactobacillus murinus species, so you don't get this conversion of indoles and this happening. So this then differentiates into these produce a lot of interleukins, which are pro-inflammatory, and this leads to inflammation and hypertension or high blood pressure and so on. So they're able to show the link between the bacteria through the metabolite into the actual final result. So they were interested in doing this in humans as well, and they did a normal salt diet, just kept people on a normal, or just took people in and then started feeding them some higher salt, and they also found that the blood pressure went up, the systolic and the diastolic here and so on. And then they looked at their gut microbiome and then looked at the lactobacillus, because they think that in the mice as well, lactobacillus was the main dropout under the high salt diet. So in the red line here, very quickly they found the numbers of lactobacillus, even though they were very low at the start, they dropped even lower. So within a few days. And then when they look at control studies where people just analyse for their gut microbiota over time, they find that the lactobacillus drops away slowly across a lot of different studies. But the high salt seems to knock these lactobacillus out much faster. So that's kind of a nice link between the mice trials and the human studies as well. So I think when I go to Bunnings on Saturday, if I can take a shot of some probiotic that's salt resistant, and it can survive my sausage, the salt in the sausage, will it reduce my high blood pressure? So these sorts of things here are coming about where it's kind of, do you modify your diet, or do you try to come up with I just want to eat what I want to eat, and I want to take a tablet to sort of deal with this sort of problem that might eventuate. But it's interesting work nonetheless. Okay, the last thing I'm going to talk about is trealose and its role in an infectious disease, which has come about in a recent study just this year. And I'm sorry, if there's food coming out soon, this might put you off it a little bit. But that's okay, it's more for me, that's fine. So I'm going to talk about Clostridium difficile infection, which is a really nasty type of infection. It's antibiotic resistant, it produces spores, it produces all these symptoms here. This is what the gut looks like with an infection with CD, and it's very difficult to remove, and it's associated with a lot of the time with antibiotics. So when people take a dose of antibiotics, it kind of strips away their microflora. If this bacteria is there, it's spore forming, so it will survive, and then it can start to grow and dominate in the gut. Once it's there, it's very hard to get rid of, and this is where things like fecal transplants are actually proving to be very successful. They're trying to put in probiotics or trying to treat with more antibiotics, and nothing really works. And what they found with patients with CD is that their gut diversity is very limited. So if they can restore that diversity just by giving an extract from a healthy person's feces back into that person, they're able to restore a diverse microflora, and it actually outcompetes this organism here and restores that. So it's kind of like a broad spectrum probiotic, I guess you could see. But it's proving to be very successful, even though it sounds very gross. So this is a huge problem in the US and in a lot of developing countries as well. And what they found is, what I'm going to focus on here, is there's two epidemic types which have really come to the fore in the last 15 to 20 years. And these guys are really virulent and increased in mortality, the kill rate they have. And this is a study that just came out 2 months ago, where they're really looking at the effect of dietary trealose, which is a sugar it's in a lot of different foods, and its role in enhancing virulence of this pathogen. So these are the two types that have come up a lot. And what they were interested in, why are we seeing these two types that are really nasty now, that are sort of just expanding across the world and becoming very common? So they were interested in looking at the metabolism of these. Is there differences between these and other Clostridium difficile strains as well? And one thing they found was that these ones could utilise the sugar trealose. I'm not a food chemist, but I think it's two glucose molecules joined together. Yep, that's about my limitation. So they grew these different strains of Clostridium difficile in different sugars. They all grow in glucose, but in trealose, only those two really nasty types grow. So this is growth up here, absorbance, and pretty much all the other Clostridium difficile types don't grow in trealose. So these new ones seem to have mutated, or something's changed with them. They've evolved to be able to utilise this sugar, which is now in our foods. It's part of our diet. So they did some whole genome sequencing. I don't want to get stuck into this too much now. But they found there was mutations in a trealose regulator in one of the types, which affects one of the enzymes that's involved in metabolizing this sugar. And this just shows that gene, it's basically the regulator is mutated in this side here, which basically makes it super sensitive to trealose, and it can switch on the trealose metabolism genes. The other one they found was using whole genome sequencing. They found there was a bunch of new genes in there which were part of a trealose transporter metabolism system. And they've done some studies to show that trealose increases virulence in mice. So when they inactivate the gene, the TreA gene, which is metabolizing it, the number of mice that die in the study are reduced compared to the wild type. And also when they feed trealose, here in the dotted line, they get a much higher mortality rate as well. So trehalose seems to be stimulating the virulence of this bacteria. The next thing is trehalose in our gastrointestinal tract, so they wanted to see that. And basically they found that when they fed mice, levels that were found equivalent to the levels that are found in ice cream and so on. This is using a sensor assay. I'm not going to go into this too much, but when you see a bar there, basically there's trehalose there that's able to stimulate the metabolism genes in these bacteria. So these levels of trehalose which were fed, and they also fed lower levels as well to antibiotic-treated mice. And that's a bit of a cheat because you can kind of strip away all the bacteria and so on. But in Clostridium difficile, a lot of the cases come about because people are on antibiotics and there's not as much competition and so on. So that trehalose, if it's there, will be gobbled up by these two types of Clostridium much more readily. That's in mice. They took 3 people from 3 humans that are on normal diets and just said, is there trehalose in the gut of a normal everyday person? And they ileostomy took some samples from that, which is during some surgery, and basically they found that 2 out of the 3 had quite high levels of trehalose. 1 person had undetectable levels. So it's not unusual that the general population has trehalose because it's found in so many different foods, and that may actually be a new food source for these types of pathogens and kind of accelerating their spread and virulence. So what's trehalose? So it's a non-reducing sugar, and it doesn't undergo Maillard reaction and acid hydrolysis. So it came about as this is a really nice new sugar that we can use in the food industry as a sweetener and so on. And before 2000, it was very expensive to produce, about $700 per kilogram. But they developed a new method, and they were able to convert from starch to produce this and reduce the cost down to $3 a kilogram, which made it commercially viable and found its way into a lot of different foods. So it was approved in the US in 2000, EU. Australia approved it in 2003, and it's on the novel food standards. And it's found in a bunch of different foods, so it's pretty common. So this paper here was then looking at, has the mass consumption of trehalose triggered the sort of rise of these 2 types of Clostridium difficile? And these are the kind of outbreaks that have happened over the years, so blurry. But there was a couple of outbreaks before that, but around sort of 2000, you can see that the number of outbreaks in all these different countries here have expanded, and every few years you get a massive outbreak of this disease. So they're really sort of pointing the finger as they're saying that, that the consumption of the mass use of trehalose has actually allowed the evolution or I guess kind of overwhelming domination of this bacteria to cause illness and sort of outcompeting other Clostridium difficile. So it's a really interesting effect of something in the diet has changed and actually triggered some quite nasty outcomes. All right, so just to finish off, and I think, as Mike sort of said, the diet significantly affects the gut microbiota, which impacts on health and well-being, and lots of different health outcomes are being related to that, as I explained during. And the diet really dictates the presence or absence and also the ratio and diversity of the gut microbiota. And I think it's not a case where you can just kind of take a probiotic now and just sort of think that probiotic's going to persist in your gut and live on and give you some health benefits and so on. It's really down to the prebiotics, as Mike said, the food for those bacteria that you really want to grow there. You need to provide that for them to hang around. And the probiotic research is really moving from things like fermentation sources, like we find this in a fermented milk and we want to use it as a probiotic and so on, to looking at microbiome studies now. So pulling out these bacteria from some of the studies I talked about and showing that they have efficacy and then possibly providing them as new probiotics. So this mining of the microbiome is an hot area at the moment. And really human clinical trial data is necessary. And if the diet is poor, I think the probiotic's not going to last around. So that bacteria, you've got to maintain this good diet, these prebiotics, which are the fuel for those, food for those good bacteria. So the future, I think, sooner or later we'll see routine gut microbiota testing, if it's not already around now, I guess. And severe situations like Clostridium difficile, where you've got issues, things like fecal transplants, where you're just providing just a broadly diverse microbiota from someone that's healthy and trying to re-establish sort of a normal population in someone that's sick. And there's evidence to suggest that in these fecal transplants in particular, but other areas where viruses that infect bacteria, so they will have, in our gut microbiota, we have just as many viruses as we have bacteria floating around there. So wherever you find bacteria, you'll find the viruses that infect them. and they can control the population, what happens to those, whether they die or break down, or how selection happens and so on like that. So it's a real community. We're kind of focusing on bacteria like we ignored bacteria 20 years ago. Now we're focusing on bacteria, but there's probably other things also that are going to make it a bit more complex as well that we need to look at. So I'm happy to, I'll finish there. I'm happy to answer any questions with Mike later on, but thanks for your attention.